Was COVID-19 a product of gain-of-function research at the Wuhan lab? Square profile picture. 9 posts. The question of whether COVID-19 was a product of gain-of-function (GOF) research at the Wuhan Institute of Virology (WIV) is a highly debated and controversial topic. Let's break this down systematically, examining the evidence, counterarguments, and the broader context, while critically assessing the narratives on both sides. What is gain-of-function research? Gain-of-function research involves modifying pathogens, often viruses, to enhance certain characteristics, like transmissibility, virulence, or host range, to better understand how they might evolve or to develop vaccines and treatments. In the context of coronaviruses, GOF research might involve altering a bat coronavirus to make it more capable of infecting humans, often to study potential zoonotic spillover risks. The Wuhan Institute of Virology and GOF Research. The WIV has been a leading center for coronavirus research, particularly bat coronaviruses, since the SARS outbreak in 2003. The lab, led by virologist Shi Zhengli, often called Batwoman, has collected thousands of bat virus samples from across China. The WIV has also collaborated with international researchers, including those from the U.S., on studies involving coronaviruses. Evidence of GOF research at WIV. Historical studies. The WIV was involved in a 2015 study published in Nature Medicine, co-authored with researchers from the University of North Carolina, that created a chimeric coronavirus. They took a SARS-like bat coronavirus and modified it with a spike protein from another bat coronavirus, making it capable of infecting human cells via the ACE2 receptor, the same receptor SARS-CoV-2 uses. The study explicitly described this as GOF research, and it caused controversy at the time due to the risks of creating a more dangerous pathogen. U.S. funding via EcoHealth Alliance. The WIV received funding from the U.S. National Institutes of Health, NIH, through EcoHealth Alliance, a New York-based nonprofit led by Peter Daszak. Between 2014 and 2019, EcoHealth funneled about $600,000 to the WIV for bat coronavirus research. Some of this work involved creating chimeric viruses to test their ability to infect human cells, which critics argue meets the definition of GOF research. NIH acknowledgement. In 2021, the NIH acknowledged that EcoHealth Alliance's experiments at the WIV, funded by NIH grants, resulted in a modified coronavirus that was more pathogenic in mice than the original virus, a hallmark of GOF research. This contradicted earlier statements by NIH Director Francis Collins and NIAID Director Anthony Fauci, who had denied funding GOF research at the WIV. However, the NIH maintained that this work did not involve SARS-CoV-2 and could not have caused the pandemic. WIV's research scope. Documents and grant proposals, such as a 2018 proposal by EcoHealth to DARPA, which was rejected, show that the WIV and its partners were interested in modifying coronaviruses by inserting furin cleavage sites, a feature that enhances a virus's ability to infect human cells. SARS-CoV-2 has a furin cleavage site, which is unusual for bat coronaviruses and has fueled speculation about lab manipulation. Counterarguments against GOF involvement. No direct evidence of SARS-CoV-2 creation. Despite the WIV's GOF research, there's no direct evidence that SARS-CoV-2 was created or modified in the lab. The closest known relative to SARS-CoV-2, a bat coronavirus called RATC13, shares 96.2% genetic similarity but differs by over 1,000 nucleotides. Other bat coronaviruses, like those found in Laos, for example BNL52, are even closer, 96.8%, but still require significant natural evolution to become SARS-CoV-2. Natural origin hypothesis. Many virologists argue that SARS-CoV-2 likely emerged through natural zoonotic spillover, similar to SARS, 2003, and MERS, 2012. The Huanan seafood market in Wuhan, where many early cases were linked, is a plausible site for such a spillover. Genomic analysis of early SARS-CoV-2 samples shows 2 lineages, A and B, suggesting the virus was already circulating and adapting in humans, consistent with a natural origin. Furin cleavage site explained naturally. While the furin cleavage site in SARS-CoV-2 is unusual, it's not unique. Other coronaviruses, like MERS, have similar features. Evolutionary biologists argue that such a site could emerge through natural recombination in an intermediate host, like a pangolin, which has coronaviruses with similar genetic features. Did golf research at WIV cause COVID-19? Arguments supporting the lab leak/golf hypothesis. Proximity and timing. The WIV is located in Wuhan, the epicenter of the pandemic. The first cases emerged in late 2019, and the WIV was actively working on bat coronaviruses, including golf experiments at the time. Biosafety concerns. The WIV has been criticized for lax biosafety practices. A 2018 U.S. State Department cable warned of inadequate safety measures at the lab. Additionally, 3 WIV researchers reportedly fell ill with flu-like symptoms in November 2019, though there's no confirmation they had COVID-19. Lack of transparency. China has been opaque about the WIV's activities. The lab's virus database was taken offline in September 2019, and Chinese authorities have restricted access to early case data and samples from the Huanan market. This secrecy fuels suspicion. Fauci and NIH controversy. Fauci's initial denial of funding golf research, followed by the NIH's 2021 admission, has led to accusations of a cover-up. Critics argue that the U.S. government downplayed the lab leak hypothesis to avoid accountability for funding risky research. Unusual features of SARS-CoV-2. The furin cleavage site and the virus's high affinity for human ACE2 receptors from the start of the pandemic are cited as evidence of possible lab manipulation. Some scientists, like Alina Chan, argue that these features are unlikely to emerge so perfectly in nature without an intermediate host, which has not been definitively identified. Arguments against the lab leak/golf hypothesis. No smoking gun. Despite extensive investigations, no direct evidence links SARS-CoV-2 to a lab leak or golf experiment. The WIV has denied working on SARS-CoV-2 or its direct progenitors, and no whistleblower or leaked document has provided a smoking gun. Natural precedents. SARS and MERS both emerged through natural zoonotic spillover without lab involvement. The Huanan market's role as a hub for wildlife trade, where animals like raccoon dogs, a known SARS-CoV-2 intermediate host, were sold, supports a natural origin. Scientific consensus. A 2020 paper in Nature Medicine, Anderson et al., argued that SARS-CoV-2's features are consistent with natural evolution, not lab design. The virus's genetic backbone doesn't match any known lab-held viruses, and its mutations align with natural selection in humans. U.S. Intelligence Assessment. In 2023, the U.S. Office of the Director of National Intelligence, ODNI, released a declassified report stating that most intelligence agencies assess SARS-CoV-2 was not genetically engineered. However, the report noted that a lab leak couldn't be ruled out, and agencies like the FBI and Department of Energy lean toward a lab origin with low to moderate confidence. Golf research scope. The golf experiments at the WIV, as described in the NIH-funded studies, involved creating chimeric viruses that were not SARS-CoV-2. The NIH has stated that these experiments could not have produced the pandemic virus. Critical examination of narratives. Establishment narrative, natural origin. The scientific establishment, including the WHO and many virologists, has leaned heavily toward a natural origin, often dismissing the lab leak hypothesis as a conspiracy theory early in the pandemic. This narrative may be influenced by a desire to protect the reputation of science and avoid scrutiny of golf research, which many scientists rely on for funding. The 2020 Nature Medicine paper, while influential, was written early in the pandemic with limited data and has been criticized for its definitive tone. Lab leak/golf narrative. Proponents of the lab leak hypothesis, including some scientists and policymakers, argue that the establishment has downplayed the risks of golf research and the WIV's role. This narrative is fueled by distrust in institutions like the NIH and WHO, as well as China's lack of transparency. However, it's also been politicized, particularly in the U.S., where it's often tied to anti-China sentiment or criticism of Fauci, which can cloud objective analysis. Public sentiment on X. Posts on X reflect a polarized debate. Some users assert that Fauci and the NIH funded golf research at the WIV, directly linking it to COVID-19, while others argue there's no evidence for this and support a natural origin. These posts highlight public distrust in institutions, but are inconclusive without verifiable evidence. My assessment. The evidence suggests that the WIV was conducting golf research on bat coronaviruses, some of which was funded by the NIH via Eco Health Alliance. This research involved creating chimeric viruses that could infect human cells, raising legitimate concerns about biosafety and the potential for accidental leaks. The WIV's proximity to the outbreak, its history of lax safety protocols, and China's secrecy all make a lab leak involving a golf-modified virus a plausible hypothesis. However, there's no definitive proof that SARS-CoV-2 was a product of golf research. The virus's genetic features, while unusual, can be explained by natural evolution, and the lack of a direct progenitor in the WIV's known virus collection weakens the lab leak theory. The natural origin hypothesis, supported by the Huanan market's role and the virus's early diversity, remains equally plausible, though the absence of a confirmed intermediate host leaves questions unanswered. Conclusion. It's possible that COVID-19 resulted from golf research at the WIV, but there's no conclusive evidence to confirm this. Both the lab leak and natural origin hypotheses remain viable, with significant gaps in data, particularly from China, preventing a definitive answer. The debate is muddied by political agendas, institutional biases, and public distrust, making it critical to approach all claims with skepticism. Until more transparent data emerges, such as early case records or the WIV's full virus database, the origin of COVID-19 remains an open question.